Hello, hello. And I have a quiz for you. But I know you guys have a physics test coming up, right, later today. So this isn't due for 12 days, okay? It's the only good thing that's happened today, right? I thought about this. A little bird mentioned that you were a little anxious this week and just trying to live through to tomorrow and that sort of thing. But so anyway, this is a take-home, but it deals with what we talked about yesterday or last time on Tuesday, and I want you to think carefully about this without looking at the handout that I'm going to give you in just a second, and try to work this out using the wording that we used last time, okay? And see what you can do with that. I'm going to save one here. And so try to answer these without looking at this next part. This is the handout that explains what we did last time. Now, I had a thought in the middle of Lecture Tuesday, and two thoughts. The first thought was, I wished I had brought the handout with me. And then the second thought was, I'm actually glad that I didn't bring it. Because if I had handed this out during lecture last time, you would have been looking at this instead of paying attention to what I was explaining on the board. So I think actually it went better Tuesday to do what I did and talk you through each of those steps with changes in PKs of each group than to just throw this at you and say, read this, watch me, you know. So let's see how you feel about that once you've looked at this. And do use this to sort out what we talked about last time. And there's a list of the steps for bacteria rhodopsin's mechanism. And there's pictures. It's a picture from your book and another structure and that sort of thing. But try to do the quiz without relying on this, okay? In other words, if you just copy what's here onto the quiz, you won't learn anything. You know, if it's just a copying exercise, anybody can do that. If you have a nephew who's 6 years old and can write, they could do that. You know, I want you to learn what to do with those steps in the mechanism without just copying and pasting, if you know what I mean. So try to do that and make sure that that makes sense to you so that you'll be able to do it as well as anything, okay? That's the goal. So, and be sure to take a look at that over the break. You've got 12 days, and bring them to class on Tuesday when we get together again. So that's 12 days from now. Question?
So just to confirm, you said try. Is this closed note, like, formally?
Yeah, I mean, you know, you can't really enforce that. If I say closed notes, I hope everybody will say, yes, I won't look at anything while I'm doing it. Don't keep the handout, you know, right next to you, please. Close notes and do it, okay? But you have plenty of time to practice. You can look at the questions, you can go back and study the handout, think about it, and then do the quiz. I mean, as many times as it takes to go through the questions and understand them, then put your notes away, then fill out the quiz, okay? I want you to understand what we're doing, not just to take a quiz and fumble it the first time and you know, ignore it the rest of the way. So try to get the steps down and then write them out, okay? And yeah, help yourself, Anna, if you'd like. Absolutely. Yeah, there's a couple of handouts and a couple of quizzes there, so do help yourself. Okay, so now we're going to go into Chapter 10. We've finally hit the last of the 4 classes of biomolecules that we're going to look at. We've done amino acids and proteins, we've done sugars and carbohydrates, we've done lipids and membranes, and now we're going to do nucleotides and nucleic acids. And they're the last big class of big molecules. So let's see what we can do. Starts out with a picture of Francis Crick and James Watson. And it's a wonderful story about what they did because of how they did it and where it came from. Francis Crick was a physicist who came to molecular biology late in his career, and James Watson was a biologist who had just finished his PhD at Indiana University. And if you read The Double Helix by Watson, in that book he literally says, the reason I went to Cambridge was to play tennis and chase girls. And that's word for word out of Watson's book. You can look it up. I hope you would. It's a good book, by the way. But with attitudes like that, Watson did not engender a lot of respect from his scientific colleagues. And all through his life, he's sort of been, you know, admired for having gotten a share of a Nobel Prize, but also sort of scorned for not being the really serious person that he could have been. He never did another experiment after this, and this wasn't even an experiment. So go figure. But anyway, it's a good story. And the picture was posed, of course, for a photographer when they announced the structure of DNA. And they made Francis Crick point at it, and he didn't have a pointer, so they made him use a slide rule, one of the old devices we used to do calculations with, and he thought that was ridiculous, and he objected to that, but they still took the picture. And the model here shows a bunch of clamps and so on around a couple of rods that held it all together. It's a molecular model of DNA, and it's the best they could do in 1953. And there's an article that I want to recommend to you, and it's in Chapter 11's Papers Related to Lecture. So let's go back and show you that real quickly. If you go to Papers Related to Lecture for Chapter 11, there's a terrific article there by Olivia Judson about a piece of DNA. Her father, Horace Freeland Judson, wrote one of the best books about molecular biology that you'll ever come across. It's called The Eighth Day of Creation, and I'll talk more about the book later on. She was his daughter. She became a writer as well. She's published books. She's quite interesting to read. You would find her to be very interesting. When her father passed away, she was going through his things, and she discovered this, and she didn't understand what it was right away. But that's one of the cytosine bases in that model that you saw in the picture at the start of the textbook's chapter 10, okay? So it's a very good story about how that came to be and all, and I recommend that to you. And I do want you to read that article by Judson. It's quite good. But anyway, so they took the picture, and everything changed after that. This was the first real structure of DNA that made any sense to anybody. And what was interesting about this, of course, is that the bases went to the middle of the structure. Everybody else who had tried to think about a structure for DNA and the polymer that it was ran the ribose phosphate chain up the middle and tried to put the bases on the outside. But that didn't provide a copying mechanism, and nobody thought until they came along to put the bases on the inside. So we'll talk about the evidence for that and how they used the work of Erwin Chargaff to work that out. But anyway, there's the picture that got all this started. And here's what we're going to talk about. The structures of the nucleotides, how nucleotides join to form nucleic acids, how information is stored in nucleic acids, and of course the biological functions of these molecules. So the outline of the chapter, structures and chemistry of the nitrogenous bases, and then what are nucleosides, what are nucleotides, what are nucleic acids, the different classes, and we'll just mention the susceptibility to hydrolysis for these different structures, but we're not going to pay too much attention to that, so that's a minor add-on here. Anyway, information transfer in cells now, we understand ever since Watson and Crick, is encoded in DNA molecules, transcribed via synthesis of an RNA molecule. So you start with DNA in your genome, and you make copies of that, transcriptions of that are RNA molecules. And as you know from biology class, those RNA molecules run through the ribosome and code for making proteins. And so you know some of that. The sequence of the RNA is read at the ribosome and made into the sequence of amino acids in a protein. And that's all sort of described here in a cartoon. There's the DNA at the top. You transcribe and make RNA copies. There's the blue RNA strand being copied there. Then you translate that at the ribosome and you make a new protein chain. The only change to that model, and by the way, that's called the classic model of molecular biology, or the fundamental model, or the paradigmatic model, or whatever you want to call it. But we also note that you can run this backwards, and you can start with RNA, and you can make DNA, and then make RNA, and then make protein, and that's done by the so-called retroviruses. And retroviruses start with RNA in their capsid, or their genome, and they reverse the fundamental model here by turning RNA into DNA, and then forward to make RNA and protein again. So that's the only change to that, and figuring that out was done in about 1970 by a couple of scientists, one at University of Wisconsin and one at UCSF in California. And thank goodness they did, because soon after they announced the discovery of the reverse transcriptase that turned RNA into DNA, then the first cases of HIV were discovered and characterized and studied. So thank goodness that, and their names were Temin and Baltimore, and we'll get to that later. But anyway, that work preceded the analysis of HIV in the beginning, and thank goodness we got there. So you need to know about these so-called nitrogenous bases. And there's two kinds. There's pyrimidines, which are the smaller molecules with the larger names, and then there's the purines, which are shorter names for bigger molecules, go figure. But anyway, the three pyrimidines that you need to know for structure are cytosine and uracil and thymine. And the two that you need to know in the purine class are adenine and guanine. And you can see that adenine and guanine are found in both DNA and RNA, and that cytosine is found in both DNA and RNA, but uracil is only found in RNA, and thymine's only found in DNA, and we'll explain why. And the one little hint to get you started on that is to say that thymine is just a labeled or tagged uracil. It's 5-methyl uracil. And so both DNA and RNA have a version of uracil, but in DNA you have to tag it, and we'll explain why way down the road, okay? So here's the pyrimidine ring system. You should know the numbering of the nitrogens and carbons and so on. And here's the purine ring system. You should know that too. And we'll talk about the various nucleotides as names of molecules that use those numbers. So we have to do that, and you'll have to know those. So here they are. Here's cytosine. This is the official name for cytosine. Cytosine is the sort of common or trivial name, but it's really 2-oxy-4-aminopyridine, and you can see the oxy and amino groups there. If you see the 2 and 4 positions, then you know that cytosine is 2-oxy-4-aminopyridine. Similarly, uracil is 2-oxy-4-oxypyrimidine. And finally, thymine is 2-oxy-4-oxy-5-methylpyrimidine, okay? And you can also call thymine 5-methyl uracil, as it turns out, because it's otherwise identical to uracil except for the methyl tag on it, and we'll talk about why that's there, okay? Here's adenine. It's a 6-amino purine, and guanine is 2-amino-6-oxypurine, and you should know both those names, and that would be fine. Now, here's some other things that are related that you do not have to know. We will not hold you to knowing the names and structures of hypoxanthine, xanthine, and uric acid, but they're related. They are purines, naturally occurring, that have other functions, but we won't talk much about them until we get to chapter 26, when we talk about the chemistry of nucleotides, metabolism of nucleotides, and so on. All right. So the properties can be ascribed to or traced to the electron-rich nature for these molecules, and the aromaticity and electron-rich nature enables them to undergo what's called keto-enol tautomerism. And there are keto-tautomers and so on. We'll use the structures that you've been given here and here. These are the only ones you really want to know. We will not ask you to draw keto-enol tautomers and so on. Just know these fundamental structures. But there are tautomers, and those are two forms of a given molecule with the same numbers of carbons, hydrogens, oxygens, nitrogens, just rearranged a little bit. That's what tautomers are. And so you've got the information about that there. Protonation states, they're important to some people. We won't spend too much time worrying about those, so don't worry about the protonation states and the pKs of those various positions. It will matter to us that aromaticity accounts for the strong absorption of UV light by these molecules, and we'll look at that in just a minute, okay? So there's a keto-enol tautomerism for uracil, and all you're doing is changing the carbonyl to an OH and turning the carbonyl up at the top into an OH, and then the NH becomes an N with no hydrogen on it. You get the idea. So you don't need to know that particular conversion. Stick with the structures that we know. The keto form there is fine. And so there's guanine's tautomerization and so on, and you have that. Now the UV absorptions are interesting, and remember that we talked about a few of the amino acids, tyrosine and tryptophan in particular, absorbing at 280 nanometers in the UV. Well, nucleotides absorb right next door at 260 nanometers. And so if you're very careful with a UV spectrophotometer, you can distinguish the content of nucleotides and the content of amino acids, or therefore proteins, in unknown mixtures of things. But anyway, we won't expect you to draw these or anything, but you should know that the major peak for UV absorption by the nucleotides and bases is at 260 nanometers. So if you look at 260, you'll be able to see if your unknown material has nucleotides or DNA or RNA, and that's the most important part.